Hello, everybody. I know this has been the burning question for quite some time about nanobots and graphene oxide in anesthetics, and we had hired a toxicologist who gave her presentation, and now we have someone to give us a presentation from a totally different perspective because I still get calls about this from lay people and from dentists. First off, I want to introduce Nova, and Nova, I have a burning question. Has anybody paid you to specifically point this research in a particular direction, or is this all your own research?
Nobody's paid me.
Okay.
Everything I do currently is unpaid.
I
So
I
I
just
a
wanted
lot of
people
it,
to know this was not in any way solicited or bent in a certain direction. Actually, I haven't even heard this presentation. So I heard about Nova and what she does, and I am going to let you take it from there because you have such an extensive background. Could we possibly start with who you are and how you got to where you are?
yes, we can. And if it's up to you, Dawn, but that would
Yes,
be slide
I'm
two.
I am going to
Okay.
share and give me 1 second. I can get this PowerPoint up here. Sorry, I should have had this done in advance. And let's tell the listeners the reason that I have the PowerPoint slides is because Nova got a new computer and
Yes,
it, it is a little
challenging.
challenge for her. So yes, so
And I haven't done PowerPoint presentations since I left the work world, so that's almost 20 years.
okay.
All right, here
So
we go. Um, so my technical expertise, I was late to returning to getting my degree. I was like 34 years old. So I was at a point where getting educated and finding out what I really did want to do professionally was very important. So I went and I got a BS in biochemistry at NC State University. I followed that up with a master's in environmental chemistry at UC Chapel Hill, UNC Chapel Hill. And then I went into my work world. And when I did that, I stayed at the university. I was lucky to be hired on there, and I worked there for until 1998 doing research. And while I was there, I was continuing on some of the things I'd done in my master's work, which is which was synthesizing my own standards and doing trace analysis, meaning that I was looking at very low levels of disinfection contaminants from the ozonation process. That ultimately ended up using gas chromatography, mass spectrometry, which was considered to be state of the art for detection and quantitation. And then when I stayed on at the university, I transitioned into a different project from the Superfund, which was looking at ethenoguanine. And this is a DNA addict of guanine. The original work was done looking at exposure to vinyl chloride, and then my PI was being funded to look at the impacts of vinyl chloride, which is, I'm sorry, vinyl fluoride, which is connected to Teflon.
mhm.
And so we were getting Superfund funding partially to get that work done. I was still very much involved in detecting and quantitating. And what is important about that is I was learned how to do proper calibration for quantitative work. So when I look at any kind of analytical work being done that's trying to identify and source and quantify what's there, I have a very strong calibration background. Um, I was also doing a lot of training at the time. I trained the master students and the PhD students that were coming through the department. And then I moved on to the pharmaceutical industry in 1998. And that was an entirely different exposure and experience because it was dealing with regulation, the regulated industry. And so that initially started out as a QA auditor, and then I developed a new corporate training program for people who were onboarding as new employees. So they got used to being in a regulated industry. And then I started doing data collection training for the proper way to use our electronic capture system known as Turbochrome. I stayed there for about 3 years, and then I had the chance to go work at first it was American Home Products and then transitioned to Wyeth Pharmaceuticals. And that was a vaccine manufacturing facility. So we were dealing with an entirely different process, and we were also dealing with different regulatory requirements, because when you are working with vaccines, you're working with sterility all through the process because of possible contaminants. And then I retired in 2008. The other thing I wanted to mention about my background is that I've also spent a lot of time in the alternative world. I've done all kinds of chelation, infusion, as well as some oral. I've done ozone therapy. I've used procaine for neural therapy. I've done traditional Chinese medicine, Ayurveda, and meditation. In 2019, I had the chance to join a health group, and we were getting products in that we wanted to assure our membership that they were, number one, what they said it was, and then we wanted to establish the quality, the purity. And so that allowed me to continue using some of my skill sets. And I still do that. And we are currently working with another oral chelator, HOPO, which is used for gadolinium toxicity, deposition disease. And because when I became aware of Dawn's concerns about what was going on in the biological dentist community, I said to her, well, I'd like to take a look at some of the data. This is right down my alley, and I'd be glad to tell you what I think is there. So, are there any questions?
No, I'm just trying to get it to advance. And even on my end,
It's not wanting to click to the next slide?
no. Isn't that interesting?
Can you just take your finger and move it up to the next slide?
No, hold on. Let's see. Turn it back on here. Okay. Let's see if that will. There we go.
Got that one. There we go. There we go. Perfect. Okay, that was slide 3. So what do we want to cover? We want to look at, I put some research results in a couple of tables so we can look at direct comparison. Then after we look at that, and that includes what was done through Jim Lundstrom's efforts as far as testing the anesthetics using different techniques. And then we have some results that were offered by independent researchers, and we'll take a look at those. After we just look at the comparison of the data results, then I want to move into some of the regulatory issues. I want to show you what a patent for the current batch of anesthetics that we're talking about, such as septicaine, such as carbocaine. I want to show you what a patent looks like, what are the things it stipulates. And then after we go through that, I want to take you through an ACS, American Chemical Society, review article. And that is for telling you what the basic characteristics of graphene oxide are and its potentials, and exactly where it is at this time and moment as far as being used in any applications. And then my thoughts as to what I'm noticing and what I think is a good explanation and evaluation of the two sets of data, the one by the traditional analysts offered by University of Colorado, and there was qPCR done, as well as some cryoscopic TEM, which is electron microscopy. And perfect, that's where we want to be, the first set of collected data. So looking at this, how I set this up was I set up what were the actual anesthetics that were tested versus what kind of results we got. And with the very first test that was done that made the biggest splash was Oroblock. And it was the University of Colorado, the first set of its data that said it was positive for graphene oxide. At the same time, the anesthetic was being analyzed for the qPCR and the cryo, TEM. What I want you to notice is that it was negative for the SV40 and the spike, the fungal, and the bacterial, and under the TEM for any lipid or carbon features. And this is important because these are actual parts of DNA that can be examined through the PCR process. qPCR stands for quantitative polychrome replication. And that is a technique where you can have a little smidgen of DNA and you can amplify it. You literally create like a copy. It's a chemical copy machine. And so if you have enough of it, you can ramp the multiplication up to a point of where you can see a signal presence, detectable presence of whatever you're looking for. And what we're seeing in the negative responses is there was not enough present in the sample they had that they could amplify it to anything. It just came out negative. And when we come down to looking at the dark field analysis that I'm primarily referring to Dr. Mihail Saya's work, this was a collection of, and we'll see the slides later on in the presentation, when she was giving her results, all we got were pictures, and they were pictures of dark field images, and there was no reference standard. There was nothing that clearly let us see what unexposed blood or blood that absolutely had no imagery like this in it. And she went through several slides showing various imagery and chunkiness, and she was claiming that things were forming over time. And the thing that was most disturbing about it was that there was no standard being run. There was no blank being run. One of the things that's very important when you do analytical work is you've got to control for your environment. You've got to control for the testing, the analytical process of your samples, and then you've got to control for the testing. So for really rigorous quantitative work, you've got to have a lot of controls put in place so that everybody can ask a question and you can say, oh, well, this demonstrates this, that, and the other in this kind of an environmental control. Or this was the condition of the machine before we started loading and running samples. This is how well we know the method that day in the lab when the person was processing the samples. We know what the condition of it was there. So that's where we are as far as the professionals who were producing data and results versus somebody who was doing something that was not what I would consider to be state of the art. So the next slide that we want to take a look at is, what is a patent for dental anesthesia all about? And it's basically the rules of the road for a product that has been through several testing phases, reviewed by various technical committees, and then it is put through the FDA process to see if it passes muster. And this particular patent was published in 2015, meaning that's when it was initiated, but I don't think it comes into its complete testing and approval until 2021. And so when you look at a sample patent, you're looking at all of what is allowed to be put in to make it. And then in this particular case, it goes through all the methods for pain relief that can be used without numbness. And you'll see that says that right in the title, right below the title, composition and methods for pain control without numbness. Next slide. So when we look at the various, we can see a local anesthetic of lidocaine 2% with epinephrine. We can look at 2%, and then we can look at various mixtures that we can do, I think, as we come across. You guys, you dentists are going to know this better than I am. But what I want to show you is all of this is stipulated. What you do with your lidocaine 2%, what you do with your carbocaine 2%, what you do with your marcaine. And I was noticing that some of the products that got tested, some had epinephrine and some didn't. So the patent would also be talking about what anesthetics, how they would be dosed, how they would be set up based on whether they had no epinephrine. Next slide, Dawn. So when we go through, oh, the other thing I'm going to say before, but that's just fine where we're at. Before we move on to the Kelly application for how you do a fill of these particular compounds, what fills the vials that you receive before you get ready to prepare them for injection. The patent also goes through the routes of administration. So it's talking about what constitutes a patient, a subject, embodiments, such as the appropriate packing materials, the kit components, single or multiple vial packages, delivery devices, package inserts, labels, instructions. All of that is locked down into a patent. So there is no opportunity, if everybody is complying with the regulation, there is no opportunity for someone to just decide, well, I think I'll do something different, because that's not allowed. And the FDA is supposed to routinely inspect various factories to make sure they are following the rules for the various products that they're making or the various types of testing that they're doing. So on this particular slide, this is the Kelly et al. Application, and this is for methods inspecting pharmaceutical containers for defects and particles. And this was an interesting patent to go through to see that it was primarily made of drawings, that it was very clear about what kind of apparatus is permissible for use with these vials, as well as what was required for the individual vials as far as size and what had to pass muster in order for them to be suitable for the fill process. So, next slide. All right. So let's talk about graphene oxide. I don't want to get too tied up in all the technical parlance, but I do want to talk a little bit about how this material, which all comes, by the way, from graphite, pencil lead. You can take sheets of pencil lead effectively, and there are ways of treating it to change it into a very linear, elongated product. It then can be modified further to aromatic structures called graphene due to trigonal planar carbons, meaning that it is a flat circle with rotating electrons. It's hydrophobic in nature, as is graphite, meaning it doesn't dissolve in water. But you can also do further chemical oxidation using things like epoxies or hydroxies or carbonyls, and you create a very different linear but ruffled structure surface. You can see in the table on the last one, GO, the carbon to oxygen ratio is 2 to 1 to 2 to 9, meaning it's hydrophilic. You can dissolve this in water. It can work into solution. This would be the form that there is more a likelihood of any that it could function on a physiological level, providing it's done in a safe manner. Next slide. So when we get into the manufacturing, I don't want to get into details because one after another, but what I want to get you aware of is that when we first started out, it was 1859, and there was the Stella Brody who got this, sometimes I feel like we're dealing with adult kindergartners here. He just decided to put a whole bunch of graphite into a bunch of horrendously dangerous acids, and then he let it cook for about 3 or 4 days at 60 degrees C, and lo and behold, he retrieved this very strange material that was very different in appearance and form than graphite, plain old pencil lead. And then once he started, it just took off like I don't know if it was a shot. I think it was 1898 when somebody decided to go further. And then people continued to keep playing with this through, I think, the 1930s. There were some things that were done. And then there were some significant, and you can see in this whole chart, there was a significant change done in 1958, 2005, 2009, but where the scientific world started taking real serious note of it was in 2010, because then somebody won the Nobel Prize for Chemistry. Once that happened, then money started flowing to the material as far as what would be some of the useful applications of it. One, I have heard that graphene and graphene oxide has a level of versatility I would never have thought a material could have. I'll list that out later for you. But I mean, it's even used to strengthen concrete, if you can imagine. Next slide. So with everything that was going on with the years of graphene oxide, they looked at UV vis, XRD, they looked at FTIR, Raman, XPS, EDX. I don't know what some of these techniques are, but people were trying a lot of different spectral techniques to try to characterize it. And the one that people ultimately ended up with was Raman spectroscopy, what is in the lower left corner. Next slide. So taking us to what we're interested in specifically, which is graphene oxide, is that graphene oxide has a lot of oxygen locked up into the structure. And the spectral characteristics that people look for are either two peaks in the upper table where it says G and 2D, or the bilayered, which will give you a D and a G and then a 2D at the lower end of the spectrum. So there is either two peaks with a single layer of graphene oxide, but in a bilayered, there will be three peaks. And the telling peak that you'll hear me discuss a couple times is sitting up in about the 2700 to 2900 2D area. If that peak does not show up, then that will say that you probably have graphene, which will be one or two peaks in the 1500 to 1300 range. But if it doesn't have that 27 to 2900, you don't have the double layered go. Next slide. So moving on to electron microscopies, a different way of looking for what might be present are these various electron microscopy methods. This has the ability, with the right type of slides being taken of the material, has the ability to blow things up by a factor of 200,000 or greater magnification. So in some cases, you are really getting down to not the electron area, but it is so close to what is actually there on a physical level that can be seen, that you can see patterns and changes in the physical appearance. Next slide. And here's the really good example of that. So when they were looking at SEM images of go with low oxygen content, this was 100 micrometers, and then that was the one on the left, and then you look at the one on the right, you can see there is this very intense ruffled quality, because if you've got all these oxygens throughout the carbon matrix, then what happens is it can't lie flat. The oxygen's much bigger than the carbon, as well as all the electrons that are cycling around in the carbon oxygen bonds, and that creates a very ruffly type of imagery. All right, so I wanted to talk about graphene oxide a little more clearly, but I wanted to really be clear about what we would not be discussing in this talk. I don't want to talk about the various things such as the nanoscale level, the high surface area, electrical conductivity, mechanical strength, solvent versatility, water desalinization, and electronics. Those are not pertinent to the talk we're at right now. Next slide. What is very relevant to what we're talking about is the biotechnology and tissue engineering that we're interested in. So the things that people are most excited about are wound closure and healing. There is a possibility that the graphene oxide could promote better antibacterial structures that help severe wounds close, resistant to drug-resistant bacteria, so to get away from having to use antibiotics. They're looking for burn victims. Could you do a complete regeneration of skin? Some people are saying, well, would it help better drug delivery? Toxicity concerns, however, concerns are exposures. If you're making graphene oxide, are there exposures in the manufacturing process and in the medical administration of dosing when people are actually working with it, not only for patients, but for the physicians and other clinicians who are regularly handling it and dosing with it, what their exposures are. And of course, if we're talking about tissue engineering, what kind of biocompatibility is there and how does it degrade in the body in a way that is not toxic? Part of the things that people are considering in biotechnology is what can we do for skin? What could we do for cartilage, bone? Is there anything that could be done in neurological applications? And dentistry is another one that people have been wondering about. Next slide. So to give you just an idea of some of the things that people have been considering and working with on the test level and wish list of what you would want it to be done, this is an example of various graphene oxide materials for wounds. And so people are looking at silver, arginine constructs. There are collagen, chitosan, I think that is another material that possibly could be used in a sandwich or in a material where you're mixing all of these together. Would these be effective for biological activity and mechanical strength? Next slide. And then there is the whole idea of tissue engineering. So here's a list of the various scaffolds people are thinking about for skin tissue engineering. It's there for reference, should any of you want to look deeper. Next slide. And then I wanted to show a picture in the review article that showed of a rodent that you can see that was a really big wound that was applied to the back. and then how they applied the nanofibrous membranes using a certain kind of geo mat. It was 1.5% geo containing membrane, and then they observed how the wound closed over about 14 days, and it closed completely. So there is a lot of things that we would like to see some additional technology available for wounds and wound healing. And these are among the things that are being researched at this point. Next slide. This is a huge list. This was in the review article that talked about all the various toxicity studies that are being done. And it starts out with really simple critters like Daphnia and earthworms, Drosophila, various flies, saline brine shrimp. They were looking at cell lines for cancer cell lines, lung cancer cell lines, mouse eyes, bacteria biofilm, jumped down to zebrafish, microcystitis, aeruginosa, freshwater cyanobacteria. So that's like pond water critters, algae, bacteria, gram and gram negative, gram positive, gram negative, E. coli, E. coli. I'm not sure what that's referring to. I'm not sure if they were testing the E. coli or looking to see if the E. coli diminished. But it did say that E. coli GO had antibacterial properties better than our GO graphite. Human cell lines and mice treated with 2,000 gram per mil high levels of graphene oxide in the testicles. And the point of this huge list is to say, this is all the toxicity testing that's being done. Nowhere do you see on that list anything that's being done to humans. So we are really in the fundamentals of doing research and asking questions of saying, for far less complex creatures than we are, what is the toxicity? And anybody that says we are to the point of trying to do anything with humans deliberately at this point is completely off the mark. So in looking at that review article, and there is the present issue that we've got, and that is, how did this all get started? Why did somebody start talking about COVID vaccines? Somehow there being something that was in them that then somehow jumped over into dental anesthetics, and how could that even happen? So as these questions were being asked, Leo Cashman of the DAMS Association had found out that there were a lot of rumors going out and about as far as these kinds of people claiming that there had been some kind of contamination. And this became such an issue that it became such an issue that they decided to ask various volunteers in the research community, would they be willing to test samples? Next sample, next slide. So what I wanted to talk about a little bit was the whole idea of this transhumanism and nanorobotics. I'm sure we all remember that when COVID came out, there were so many conspiracy theories. People did not see it as a simple outbreak that became global. What people thought were that there were lab leaks. Some people were very upset about the fact that there was a lot of companies that were insisting on vaccinations. Um, there has been this whole concern about things moving so quickly that were we possibly getting into that whole point of, is there a grand plan to do something like transhumanism? Are there little minuscule bots that could self-assemble inside human blood? And then would these infected humans somehow start becoming transhuman? Next slide. So I looked it up just to see what the appropriate definitions are. And I thought this was a real interesting one I found in Wikipedia. And it was talking about nanomachines. And it said, well, they are in the research and developmental phases, and there are some perhaps primitive molecular machines and nanomotors, but the reality is there's nothing there that anybody is doing in a coordinated, tested, verified level. And so as people got deeper and deeper into talking about what were all the possibilities, there was nobody who was standing up in those particular discussions and saying, based on what? Where is any of this coming from? The definition of transhumanism is that the belief that humans should strive to transcend the physical limitations of the mind and body by technological means. And there are people who do espouse such a belief. And then there is a third piece of that, which is it's a philosophy favoring the use of science and technology, neurotechnology, biotechnology to overcome the human limitations and improve the human condition, whatever that means. Next slide. And I think this takes us to any of us who remember the second Star Trek series, and that was the cyborgs. So this is, to me, as I was looking through all of this, I said, yeah, that's where we're at. We're talking about cyborgs. Next slide. So when the DAMS letter came out in December of 24, there was a letter put out of concern talking about what clients and patients of dentists were saying and the concerns they had. And we can thank Jim Lundstrom for creating a repository for all the communications there that followed. It's a great record. Next slide. So there were about 30 individual companies and professors and individuals associated with the initial graphene oxide concerns and presentations, but only a few responded. And the people that completed testing were in the tables that I showed at the beginning of the talk. The medicinal genomics, that's the place that did the qPCR, and the and looking for the pieces of the COVID vaccines as well as bacterial and fungal contaminations. And then there was the University of Colorado nanoimaging, which did the cryo transmission electron microscopy. The thing that I thought was interesting was nanoimaging services. They got 10 aliquots and for some reason only tested one. I couldn't understand that, why they didn't test more of those samples. And then the last one is Anna Maria Mihalcea, the one who did the dark field microscopy. Next slide. Where a lot of this started getting stirred up was in 2023. This is where we get into a lot of the citizen actors. The big one that everybody seemed to be coalescing around was La Quinta Columna organization run by a Ricardo Delgado. And he was running a lot of meetings, as well as he has a regular publication that comes out of his, what is it, La Quinta materials. And they were among the first, La Quinta Columna was among the first to show the wire images we'll see in a moment that were coming from dental anesthetics from someplace in Scotland. So somebody had dental anesthetics, and why they submitted the samples, it wasn't clear. It was just the imagery that was there. Clifford Carnicom, who has a really interesting background, was also involved in some of these meetings initially. And then Dr. Nixon of Nixon Labs in Australia said that they absolutely could confirm that there was this type of thing going on as far as contamination of the vaccines and a global dominionist agenda. That is part of what La Quinta Columna talks about. They talk about the EU Graphene flagship, which is a 10-year program with 170 academics and industrial partners from 22 companies. And they say there's no question that the transhumanism is an integral part of the graphene flagship. Next slide. The other thing is that back in the meetings around April of 2023, there were claims that Go was in COVID-19 vaccines. I'll address that in a moment. And then Delgado is mentioned. His training is in biostatistics. He is not a lab analyst or a researcher in that way. The news source mentions the Graphene Flagship, which is a consortium of several EU countries, which they refer to as the Fourth Reich, collaborating on researching. And then Delgado claims he found graphene oxide on face masks and PCR swabs. La Quinta Columna's channel features other videos of geo and flu vax, and then it mentions various research investigations, but none of the publications, if there are any, are cited. So we have no literature, we have no peer review results that are being compared to. We just have a lot of claims. At no point, by the way, in the imagery that they post, do they say exactly what we are looking at. They say we're looking at geo in something, but they don't talk anything about what kind of analytical techniques they're using. Next slide. So here we are. This is some of the imagery of what we all call the wires. I don't know what we're looking at. They're just, it's there. And they say it's from Scottish dental anesthetic samples. I don't have a reference standard. I don't have a blank slide to compare against. I just see images, all kinds of different ones. And in some cases, then there's a story that's attached to whatever image or whatever film. Sometimes they're films that run for 2, 3, 10 minutes. Next slide. So then that brings us to Dr. Anna. Dr. Anna is a really interesting personality. I call her a hybrid persona. She is a medical doctor. She's medically trained. She's an internal medicine with a PhD in pathology. And she was a full-time internist at a VA satellite clinic for, I think, 10 years. She was also, no, it was the Geriatric Institute. She was medical director of a geriatric institute for several years. And then in 2019 was certified as a chelation practitioner. She has a website. This is what I'm showing you here. These are all pictures that were taken from her website. And she transitioned to integrative medicine in 2018 and founded True Blue Medic in 2020. Her integrative experience and practices and work includes psychic surgery. She's a Reiki master, Chinese acupuncture, near-death experience, out-of-body experience, and then she's been trained at Ramtha School of Enlightenment, including remote viewing, telepathy, telekinesis, and she is a graduate Gateway Voyager. She has written 3 books. Transhuman, notice the title, Transhuman, copyright 2023. There's volume 1, which is the real COVID-19 agenda. And there's volume 2, overcoming the global depopulation agenda. Her 3rd book in 2021 was Light Medicine. I've included links here if anybody wants to look at some other material. Next slide. So I wanted you to see some of the presentations she has done, their interviews on Z Media. This is a subset of Alex Jones' InfoWar Productions. Dr. Anna includes, this was off of her website, she includes all of her YouTube internet interviews. So it's there for anybody to see. She is most personable. She's very personable. I saw her with one interview where she talked about growing up as a child and playing in her mother. Her mother was a physician, playing in her mother's office because there was no child care when she was young. And so she was surrounded by a traditional medical environment, but apparently had felt some kind of spiritual stirrings during her, I guess, mid-teenage years. She pursued getting an MD, and she has a PhD in toxicology, no, pathology, a PhD in pathology. So she has been rigorously trained, but has chosen to go the alternative route. So she has made a point of, next slide, capitalizing on the aura block, the graphene in the aura block anesthetic that was found at the University of Colorado. And she did a presentation on that, saying that this is absolutely confirmation that we are seeing graphene oxide in human blood as a result of the dental anesthetics from COVID vaccine. We did not see those same results in later analyses of dental anesthetic, but I didn't see her making an issue of saying, well, maybe not, after those 2 results came out of the University of Colorado. Next slide. Oh, Dawn, yeah, there you go. So this is the first series of the 3 that I allude to in the table. And she just talks about the images, and she says that this is what I see in human blood for comparison. And then below are more images of the crystal and microchips that are developing. That's her take on it. Next slide. And then she talks about how some big things are forming. Darker set of images look like big chunks, either surrounded by globular light or a diverse background of light and dark. And the final image is of a bent wire. Next slide. The bent wire, where she says we see one of these spheres developing. What sphere? A sphere that does what? Do you have an example of a sphere that isn't trapped in one of these dark fields, or a standard? Do we have a point of reference where we can make some sense of the imagery we're seeing other than a story, a narrative that the presenter is creating? By the way, all this stuff is published on Substack, which is perfectly legal to do. That is a non-censoring outlet for various articles and videos, interviews, whatever. And this is how she releases her information to the world. Next slide. So this was the particular episode where she is talking about the University of Colorado results, and that we're finding nanotech found in vaccination, also observed in dental drugs. Dr. David Nixon from Australia confirmed microchip development from dental anesthetics. And Dr. Lindbergh from Illinois also found nanotechnology in dental anesthetics. So we have these proclamations, these statements being made, and the nice thing about doing this kind of stuff when you don't have any rigor attached to it is that it's your say-so. And if somebody says you're wrong, and how do you know that is what is going on? They've got an easy out from in their defense, which is, well, there is nothing. And, you just have to feel it out. Does it make sense to you? Does it feel right? And that's not how science works. Next slide. This was another one about the breaking news on the graphene confirmed in dental anesthetic. Next slide. The last thing I wanted to show you were these two slides that are on her website, and it just gives you an idea of all the various titles, what they're talking about. And then, of course, because Dr. Anna is a physician, then she is offering all sorts of ways that we can dismantle the nanotech within our bodies. Next slide. And then more interviews and the war for humanity, nanoterror, AI nanotech of insulin, horrifying discovery in unvaxxed blood. And that's it as far as slides. I think the thing that I take away from all of this is that the reason why medical care and dental care has advanced as far as it has is because we do have tight regulations. The regulations come from trained researchers and analysts and committees formed from those trained professionals There are standards that they have to meet in order to be published. And there are standards that when you're in the process of trying to get your method approved by the FDA, there are rigorous standards you have to pass. The idea that somehow little bits of vaccines would somehow cross over into dental anesthetics is ridiculous, partly because the two different types of manufacturing facilities, one is completely different than the other. I haven't gone and done a verification of this, but I do believe that if a company were to do vaccines, as Wyeth did, as well as some solid dose, those two product lines would have completely different building sites and completely different regulations. So there could not be any crossover unless there was deliberate sabotage. And if we're going to talk about that, then we're back into the country of conspiracies. When you are making vaccines of any kind, it has to be in a sterile environment. However, if you're going to make something like a pharmaceutical cream, it has to be sterile, but the requirements for doing that in a solid dose facility are basically regulated to a discrete suite where microbiologists work. You wouldn't have to have that kind of rigor throughout your whole facility. The other thing I wanted to point out to you is the whole idea of not just validating a method and how you actually make the soup. There is the manufacturing facility validation. So when you're making something like a vaccine, and in our case with Prevnar, when I was working there, it was 7-valent, and then it stepped it up to 13-valent later on. You had a separate fermentation process and tank for each different valent. So if you had 7 valents, you had 7 different places that you would ferment for those particular meningitis strains. And it can take a year to 2 years for new facilities to be validated to a point of where they're allowed to make product that can be sold. So you can make your test batches to make sure that you've gone through and carried through the whole process. And it's meeting the marks every step through the process and conclusion, but you can't sell that material. That's just testing material. So it takes a lot of money and a lot of time and a lot of expertise to even make manufacturing facilities acceptable for something like vaccine manufacturing. And then the last thoughts I have for you. Research. Research this day is driven by money. Somebody's got to pay for it. I hear in my groups all the time, well, I wish somebody would just make such and such. And my response, which they get tired of hearing, is, well, who's going to pay for it? If you can't get money from the government to do that kind of funding, then you have to find a profit-making corporation that's willing to put the money in. And if you're talking about something different, like, say, mercury toxicity, removal of that metal from people's bodies, the fact of the matter is trying to get a product approved in the United States, and we have someone who's doing that right now, has been trying to do that for at least 10 years. Somehow it's very difficult to get the approvals. A lot of times these are orphan drugs. They don't require quite the level of validation because they're never going to be big money makers. But you got to have money in order to do this. When you turn your focus to the alternative medicine world, it's an anecdotal world. Even though we know things absolutely do work, it's always anecdotally based. We've had some superb chelation physicians the last 30, 40 years. By the way, they're my generation or a little older, and they're starting to die off. Young people are really not getting into this type of work. You don't make a lot of money doing it. You do it because you love it. But if you are living in a world of anecdotal and it's word of mouth, and there is not the scientific rigor backing a lot of what we would like to ideally see because there's no money for research, then you are also living in a world of rumor and a rumor of, gee, I had a really good result from this. You need to try that. And I've lived in that world. And so there's been a lot of things I've tried because I know it's been around for a while. So if we live in a world that is never getting the funding it needs for the scientific rigor we would all like to see, then we have problems like conspiracy theories that can really poison the well. And so I'm hoping that this presentation, while it's a different technology, it's different training, we're all professionals. And all of you dentists are very, very trained. And then on top of that, you just have your lived experience of working with your clients, your patients. And I think it's perfectly okay to be able to push back to your people and say, it's your choice. You know, you don't want to use dental anesthetic. Between you and me, I think that's nuts. This is me talking to you. That's crazy. I couldn't do it. But you can offer that to their clients. But I think it's nuts myself. But there's just no reason to believe that. There's just absolutely no reason at all. And I'm hoping this gives people more comfort with being able to stand up and say, you know, no, that's just a bunch of malarkey. There's no there. There is no appearance of graphene oxide. And we're in different generations now, by the way, of vaccines, COVID vaccines. This is several years after the beginning. I'm sure whatever they're doing is they have figured out better ways of optimizing it. Because remember, this was an emergency approval back in 2020. They're going to be that much tighter in their production, and they're going to be that much more rigorous. And I'm sure they are at the point now, and it could have been 2-3 years ago, that they have locked down their method, and nobody is sneaking anything out of or into those facilities. And I think that is as much as I have to say about it. And if there are any questions, I'm glad to entertain them.
Well, I know from my perspective, it's been very enlightening about the amount of research that you went to try to educate. Let me see if I can recap some of this. There are some individuals, especially using dark field, that found images that they could videotape or take pictures of without showing what a blank slide was as a reference, that made claims that this is what it was without any definitive testing to prove that's what it was.
Yes.
Okay. And in all of the research that you have done, which again, thank you, I wouldn't have even known where to look for some of this stuff. And just the fact that you were told, this is the question that we have. Can you tell me, in your opinion, is it true or not? In all of your research, you found no graphene oxide and no nanobots in any dental anesthetics.
No. Dental anesthetic is clean. Whatever was seen in the first confirmation at the University of Colorado, it did not have any confirmatory testing by, say, another Raman specialist. So like if they had had two different researchers who had tested the same lot, aliquots, another thing they could have done is they could have taken several aliquots and dried them down together and say, okay, we took 5 of these and they dried them down and then processed them. That would be another thing you could do. There was no calibration curve. So we don't have any way of, there's no other confirmation testing other than, because OraBlock was also tested by the genomics lab. And they're saying there's no there. There's no spike. There's no SV40. And there was no bacterial or fungal presence using the PCR technology.
And I know that you don't know the toxicologist that we were working with. She, in her presentation, said it's really hard to find anything that's made with water nowadays that doesn't have some level of microplastics in there. But
I think that's so important. It's everywhere.
Yeah, and it's not like we can avoid that. I think she even said that they took samples from just the most remote place in the middle of an ocean, as deep as they could go, and they could still manage to find. We're living in a toxic environment, and the bottom line is we do the best we can with the information that we have. And dentistry is an incredible example of how things change, and we try our darndest to improve them. So the materials that we are using now are different than the materials we used to use. And as research continues and we find out that there's something better, then we try to move on with that. But gosh, Nova, this presentation is so thorough